We saw that the cells of the trunk neural crest cells that take the dorsolateral pathway give rise to melanocytes, whereas those that go through the ventral pathway and migrate through the sclerotum, most of these become neurons and glia, with some exceptions. The types of cells that these ventrally migrating cells become depends on the factors that they encounter in their migration, but also on the receptors they express on their cell membranes. And this is a different representation of this pathway, which shows the dorsally migrating neurocrest cells that you can see become the pigment cells of the skin. They migrate between the future epidermis, which is shown in the blue, and the dermis, which is not shown clearly in this figure. Now, the neurocrest cells that take that ventral pathway are going to migrate through the sclerotum, and those that remain at the region of the sclerotum will give rise to the dorsal root ganglia. In this image, the left-hand side shows the embryo less developed, and the right-hand side shows it more developed. So the cells that remain here at the dorsal root ganglia region give rise to neurons and glia of the dorsal root ganglia, which are the sensory neurons of the peripheral nervous system that will connect to the spinal cord. The cells that continue to migrate more ventrally are going to migrate to the levels of the aorta, and they give rise to sympathetic ganglia. So these are ganglia of the fight or flight response. And those that continue in their migration even further are going to become the cells of the adrenal medulla. So this is the developing adrenal medulla, and this is the developed adrenal medulla that you see here. At the vagal and sacral levels, the cells that migrate ventrally become the parasympathetic ganglia or enteric neurons of the gut. For those cells that remain at the dorsal root region, they give rise to the sensory nerves that are going to bring sensory information to the spinal cord. But cells that undertake that ventral pathway of migration have the capacity to differentiate into different nervous system cells depending on the factors that they encounter and the receptors that they express on their cell membranes. So those cells that express the receptors for the factors Wnt and neurotrophin, which are secreted from the dorsal aspect of the neural tube during development, will remain at the region of the dorsal root ganglia and give rise to the neurons and the glia of the dorsal root ganglia. The decision to either become a neuron or glia is based on juxtacrine signaling, so that those cells that express more delta become neurons and those that express more notch become glia. Cells that do not express receptors for Wnt and neurotrophin continue to migrate ventrally and contribute to both sympathetic ganglia and also cells of the adrenal medulla. So this means that at the trunk neural crest cell level, the cells that do not express the Wnt and neurotrophin receptors, and these factors are secreted by the neural tube, will give rise to different cell types that are involved in the fight or flight response, specifically the sympathetic ganglia and the endocrine cells of the adrenal medulla. These different cell types secrete epinephrine and also norepinephrine, which are involved in that fight or flight response. Another group of neural crest cells that migrate ventrally and do not express receptors for Wnt and neurotrophin are the vagal and sacral neural crest cells, which become in part the enteric neurons of the gut. So these are the neurons that will populate the gut and are important for peristaltic movement. The fate of the vagal and the trunk neural crest cells has been shown to be labile. So these cells are not committed at the time of their migration. So if we were to exchange the vagal crest with trunk crest, we would see a reversal in cell fate, so that some vagal cells would acquire the trunk phenotypes and vice versa. So that tells you that the differentiation potential of these cells is highly dependent on the factors that they encounter along their migration and in their location of their final differentiation. In addition to being important for the differentiation of neural crest cells, paracrine factors that the cells encounter are also important for determining their migration pathways. So for example, the trunk neural crest cells are prevented from entering the gut by factors that are produced in that region that block these cells from entering that pathway. These factors are not produced at the vagal and sacral levels, so the cells that arise from the vagal and sacral levels can invade the gut. And in addition to that, there are paracrine factors at these levels that are produced by the gut that promote the migration of these cells into the gut. One of these factors is the factor GDNF, or glial-derived neurotrophic factor, which is not only important for attracting the neural crest cells to the gut, but also for their growth, their migration, and differentiation. Now, the vagal and the sacral crest both express the GDNF receptor, and remember that the GDNF is going to be secreted by the cells of the gut, the mesenchyme of the gut. So the vagal crest and the sacral crest, which are migrating into the gut, express a receptor for GDNF so they can migrate in response to GDNF. However, the vagal cells, which are the ones that are located near the region of the neck, express higher levels of GDNF receptors, so they are more invasive than the sacral crest. For that reason, the majority of the gut will be colonized by vagal crest cells, whereas the sacral crest, which express lower levels of GDNF, are going to colonize the distal portion of the gut, which is known as the hindgut. Now, megacolon results from defects in the migration of either vagal or sacral neural crest cells into the gut, so that there is a lack of neurons, enteric neurons, in the distal portion of the gut, because the distal portion of the gut is the last portion to be populated. Now, although the sacral crest contribute to the hindgut, the defects that are seen that result in megacolon typically result from the lack of cells that come from the vagal crest. This is because these cells that come from the vagal crest, they are also going to be contributing to the hindgut, but they have to migrate a longer distance. So if these cells are not able to migrate, there aren't enough neurons in this region to produce the appropriate peristaltic movement. Experiments in mice have shown that lower levels of expression of the GDNF receptor, which is known as RET, in combination with mutations in other genes that are important for the survival, proliferation, migration, and differentiation of the enteric neurons, leads to megacolon, which is basically the expansion of the distal part of the colon that results from the lack of peristaltic movement and the accumulation of feces in that location. Now that we've discussed the factors that are important for the specification of the cells that take the ventral pathway, let's look at the factors that are important for the specification of the trunk cells that take the dorsolateral pathway and give rise to pigment cells. In vertebrates, all of the pigment cells, except for the cells of the retinal pigmented epithelium of the eye, are derived from the neural crest. And recall that these are the cells that take the dorsolateral pathway, so migrating right underneath the epidermis and above the dermis. Now, it turns out that the cues that prevent the ventrally migrating cells from entering the posterior somites, so the cue that inhibits the migration of these cells, which is one of them is ephrin, actually promotes the migration of the dorsolaterally migrating neural crest cells along the dermis, so right underneath the epidermis. So in the case of these dorsolaterally migrating neural crest cells, the ephrins promote rather than inhibit their migration. Several factors, which include both transcription factors and paracrine factors, have been shown to be required for melanocyte development. These include the transcription factor MITF, which is considered the master regulator transcription factor for melanocyte development because it's needed for the survival, migration, and the differentiation of these cells. There are certain breeds of dogs and in horses in which heterozygosity of MITF causes the random death of melanoblast. So that's why you see this spotting phenotype. In humans, mutations in MITF as well as mutations in other factors that are important for melanocyte development typically lead to the appearance of a belly spot, and this is also seen in the mouse. And the reason for this is because remember that the melanocytes arise from neural crest cells which delaminate from the dorsal aspect of the neural tube. So these cells are migrating from more dorsal locations to more ventral locations. So if there's a mutation that causes the death of these melanocytes, fewer of them make it to the belly region, and this is why a belly spot appears.